So in previous class, we discussed in post-fertilization structures, that is zygote and embryo, zygote and endosperm. So here, in every angiospermic plant, there is presence of double fertilization. So double fertilization is nothing but a first male gamete fused with the egg cell. It is called syngamy. That is first fusion of a male gamete and egg cell. It is a fertilization. It leads to formation of a diploid zygote. In second fertilization, that is second male gamete fused with a central cell. That central cell is diploid. It consists of two nuclei. As fused second male gamete with that is polar nuclei, it leads to formation of a triploid primary endosperm cell or primary endosperm nucleus. So those primary endosperm cell, it leads to formation of endosperm. As we discussed, the endosperm is a nutritive tissue as it supplies the food for developing embryo. So there are three types of endosperms we discussed in last class, that is nuclear endosperm, cellular endosperm, and third one halobial endosperm. So these three types, based on that primary endosperm cell, it is divided, the nuclei of primary endosperm cell, that nuclei divided, when the nuclei divided by the formation of cross wall or not. If in nuclear endosperm, the nuclei divided, it is not followed by cross wall formation. As like there is formation of a number of nuclei, a mass of nuclei formed in that endosperm. So as it is called as also free nuclear endosperm. Free nuclear endosperm. So there is the example of a dental coconut water. It consists a number of many nuclei. So after later those nuclei may form, that is the cross walls. It leads to formation of cellular endosperm. As it is the example of that is coconut meat or coconut kernel. So it is later these free nuclear endosperm converted as a cellular endosperm. The third one is intermediate to these, that is free nuclear and cellular endosperm, intermediate type. So because in halobial endosperm, the primary endosperm cell divided first by formation of cross wall. It leads to formation of two cells. But later on, in these two cells, one cell nuclei may divide it without cross wall. The nuclei of one cell divided without cross wall. But another cell, it may retain the same. Maybe that single nuclei present in that cell may divide certain times and it will be stopped. So as it is halobial type of endosperm is intermediate of those free nuclear or nuclear endosperm and cellular endosperm. So the next embryo. So here the embryo formed in embryo sac. In the embryo sac, the egg cell which is present towards the micropylar end. It is the micropylar end, it is the chalazal end. Because the egg cell present towards the micropylar end, as the embryo development takes place in the embryo sac towards the micropylar end. Primitively, these egg cell that is fused with first male gamete, it leads to formation of zygote. So, you clearly seen in the diagram, it is the zygote. So, in this diagram, it is a little bit inverted. So, it is the micropylar end, it is the callosal end. So, these are degenerated antipodals. That is, after fertilization, all these three antipodal cells are degenerate, self-disintegrate. So, the zygote present towards the micropylar end. As the embryo developed in the embryo sac towards the micropylar end of that microscope, that embryo sac. So, these zygote, see here, the zygote which divided two cells, but it is unequal. That is basal cell and terminal cell. Basal cell and terminal cell. It is the basal cell, a large basal cell and a small terminal cell. Once again, these two cells which are divided, the basal cell which divided to form 6 to 8 cells of entire structure, what we are called as, that is suspensor cells. That is a structure called, that is totally suspensor. It leads to form of like a pastoria-like structure. It helps to, that is absorption of food in, that is later developments. But another one cell, that is the terminal cell. These terminal cell, it can take part in, first they divide vertically and later it divide horizontally. A mass of cells are formed. Those terminal cell, it divide to form a mass of cells. It is just like a spherical shaped structure as it is a globular embryo. So here the embryo, it is the first stage, that is pro-embryo. After the pro-embryo, which developed as globular embryo. So it is nothing but the terminal cell divided vertically and horizontally to form a globe shaped like structure, that is spherical shaped structure, that is a globular embryo. So once again, the cells of these globular embryo which divide, it leads to formation of these globular structure. It leads to slowly, that is a little bit heart shaped. So as you recognized here, so it is the heart shaped embryo. That is the next stage, that is heart shaped embryo. These heart shaped embryo which completely developed as mature embryo. Like the different stages of embryogeny. These embryogeny of both monocots and dicots is same. The early stages of that is zygote into development of embryo, that is same. But structurally the monocot and dicot embryos are little bit different. So those, just we know that the structures of monocot and dicot embryos. So first we discuss, that is a dicot embryo. So in dicot embryo, there is presence of two cotyledons. An entire embryonal axis, it consists two cotyledons cotyledons, plumule, radical and root cap. See, in the region, it is a structure of a dicot embryo. The entire dicot embryo, there is two cotyledons. Here, the cotyledons originated from the tip portion. So, in these embryonal axis, the level above the origin of these cotyledon. So, ಈ cotyledons ಇಲ್ಲಿಂದ originate ಆಗಿದೆ. So, where the origination place, ಈ ಸ್ಟ್ರಕ್ಚರ್ ತಗೊಂಡಾಗ ಇಲ್ಲಿಂದ ಮೇಲ್ಭಾಗಕ್ಕೆ the portion of embryonal axis above the level of cotyledon is called as epicotyl. Epicotyl is nothing but a portion of embryonal axis above the axis above the origin of cotyledons. So it is called epicotyl. So these epicotyl, it consists of two cotyledons and the plumule. The plumule is nothing but that is stem apex. The stem apex. In the stem apex, there is presence of certain leaf primordia, a small primordial leaf structures. So where it is in future which developed as a stem and the parts of stem. After a portion of embryonal axis below the origin of cotyledons. ಈ embryonal axis ಅಲ್ಲಿ cotyledons origin ಆಗಿರತಕ್ಕಂತ ಕೆಳಭಾಗ. It is what we are called as hypocotyl. Hypocotyl. So hypocotyl is nothing but a portion of embryonal axis below the origin of cotyledons. ಎಲ್ಲಿ cotyledons origin ಆಗಿದೆಯೋ ಅಲ್ಲಿಂದ ಕೆಳಭಾಗಕ್ಕೆ hypocotyl ಅಂತ ಕರೀತಾರೆ. So the hypocotyl, it consists, that is a radical part and a root cap. So here the radical which developed as root system or roots. So you know that the radical of dicot embryo which developed as taproot system. The radical of dicot embryo which developed as taproot system. So there is presence of mother root, secondary roots, tertiary roots, like a number of branches are there. So these radical which ended with a small root cap region, the root cap which covered the tip portion of radical. So while it covered the tip of radical, it protected the very meristematic cells present here, which are actively in cell division, as it is very sensitive. So that's why those a sensitive part, it is protected by these root cap. The root cap is also helps to the root entered into the soil. So as the dicot embryo, it consists two cotyledons, a plumule or stem apex. The hypocotyl consists radical, it is originated or it is developed as root. It is also called as a root apex. The root apex is correct. So as it is ended with or protected by a cap-like structure called root cap. So these just the dicot embryo. So after monocot embryo, so structurally different. The development of early stages is same both in monocot embryo and dicot embryo. See, as a structurally, the monocot embryo is of different from dicot embryo. In dicot embryo, there is presence of two cotyledons. Two cotyledons in dicot. So, but here there is presence of only one cotyledon. It is the cotyledon of monocot embryo. So here also, what the portion? It is the origination portion. The embryonal axis of embryo, that is embryo, where the origination of cotyledon, above the origination of cotyledon, that is what we are called as epicotyl. So below that is hypocotyl, it is termed. So here only one cotyledon present in the monocot embryo, a single cotyledon present in the monocotyledon embryo, it is called as scutellum. Single cotyledon of monocotyledon embryo, that single cotyledon it is called as scutellum. So in dicot embryo there is presence of two cotyledons, those which are present in lateral sides, both lateral sides. But here the monocot embryo consists a single cotyledon, it is present one lateral side, either any one lateral side. So the origination of above these it is plumule. Above the origin of cotyledon, the portion of embryonal axis called plumule. The plumule, it is a stem apex. The stem apex it consists of certain leaf primordia. Primordia is nothing but a young leaves, very young leaves. So just you see any seed, it is soaked in water in whole night. So just you open that seed, you see in the dicot, easily it is opened. So you identified a small leaf primordial structures. So those which are developed as a leaf in the next seedling pod. So those what we are called as leaf primordia. So here there is presence of leaf primordial structures here. It is entirely what we are called as shoot apex. But these plumule or stem apex, a special characterization in the monocotyledon, it is completely covered with a mass of cells, that is a sheath called, that is coleoptile. See here, it is a structure, a sheath, a layer, it protects the entire leaf primordial. It is also leaf primordial, that is shoot apex. It is covered with a layer. It is what we are called as coleoptile. The coleoptile is nothing but a sheathy layer. A sheathy layer which cover plumule, which completely cover the plumule part. So that is coleoptile. So the portion of embryonal axis below the origin of this cotyledon, it is what we are called as hypocotyl. The hypocotyl, it consists same that is radical and root cap. Radical and root cap. But these here also, as like in the epicotyl, the hypocotyl parts of root cap and root that is radical, it is completely covered with covering sheath is called, that is polyorhiza. 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 Polyorhiza is a covering sheath of radical and root cap or hypocotyl. It is also which cover the root apex and root cap part in the embryo of that is monocot. So it is the structure of monocot embryo. It consists of plumule part and radical. Next step. Seed. What is seed? Seed is the final product of sexual reproduction. In flowering plants. Because the end of sexual reproduction, it produces seeds. As the ovules which only developed as seed. When their ovule is developed as seed after fertilization, hence seed is also called as fertilized ovules. Seed is also called as fertilized ovules. So it is nothing but in the ovule there is presence of embryo sac where the fertilization, double fertilization takes place. That ovules are developed as seeds. That entire ovule present inside the ovary. As we know that the bulged portion of pistil, the basal portion of pistil, it is called as ovary. That entire ovary which developed as fruit. The ovary developed as fruit, the ovules developed as seed. So here, the seeds are of two types. The seeds are of two types. Two types of seeds based on presence or absence of endosperm. Based on presence or absence of endosperm, there are two types, that is non-albuminous or exalbuminous seeds. So the name also indicates non-albuminous seeds, that is nothing but the endosperm is completely consumed. Completely consumed by embryo during embryo development. During embryo development. So these seeds are called as non-albuminous seeds. So the endosperm, it is a nutritive tissue, as already we discussed. The endosperm, it is consumed by embryo. During embryogenesis, the embryo which absorb the nutrients from the endosperm. If that endosperm is completely consumed by that embryo, the endosperm part is completely absent in such seeds. So such seeds are called as non-albuminous seeds. Example: pea, groundnut, etc. So the second type is albuminous seeds, which are also called as endospermous seeds. Endospermous seeds. Albuminous seeds are, that is endospermous seeds. It is nothing but the endosperm is not completely consumed by the embryo during embryo development. Means certain amount of endosperm is remaining in the seeds even after completely development of that embryo in the seed. Such seeds are called as, that is, albuminous seeds. So the endosperm is not completely consumed by the embryo during embryo development. Certain amount of endosperm is remaining. Certain amount of endosperm is remaining in seed. Certain amount of endosperm is remaining. Such seeds are called as albuminous seeds. Albuminous seeds. So it is depending on presence or absence of endosperm in seed. The seeds are of two types: non-albuminous seeds and albuminous seeds. So the albuminous seeds, the example, most of all cereals, that is wheat, paddy, barley, etc. Most of all cereals. So those seeds are called as albuminous seeds. In the albuminous seeds, certain amount of endosperm is remaining. As you see here, there is presence of a monocote, a male seed. In the male seed, there is this portion, it is completely endosperm. The endosperm is remaining in the male seed. So, as it is called as albuminous seeds or endospermous seeds. So, not only remnant of that is endospermic seed, in certain seeds, remnant of nucellus. So, we discussed that the nucellus, it is a nutritive tissue, it is present surrounding the embryo sac. As the nucellus is also a nutritive tissue. Nucellus is a nutritive tissue which supply the food for embryo. So sometimes the certain amount of nucellus is also remaining. Such remnant of nucellus is called perisperm. So very important, perisperm is nothing but remnant of nucellus. That is what we have called as perisperm. For example, black pepper, even beet. So these seeds in which the nucellus is remaining, the remnant of nucellus is called as perisperm. So in black pepper, certain amount of nucellus is also remaining. So that is what we are called as perisperm. So here, at the time of the complete ovule, ovules which are developed as seeds after fertilization. As we know that is the ovule, it consists two integuments. The ovule consists inner and outer integuments. Most of the ovules consist two integuments, such ovules called bitechnic ovules. Only certain ovules consist a single integument, that is unitechnic ovules. So these integuments of ovule which developed as seed pod, that is outer integument which developed as testa, inner integument which is developed as tegma. That is the seed pod, it consists two layers, that is testa and tegma. Testa and tegma. So it is easily differentiated in the seed pod. Why you remove the seed pod of a seed? It is little bit various color, various nature. You differentiate easily, as in tamarind seed, as in groundnut seed. It is easily differentiated. So that is two integuments which are developed as, that is seed pod. The entire ovary, it is developed as fruit. The outer wall of ovary, which is developed as outer wall of fruit. The outer wall of ovary, it is developed as outer wall of fruit. So this outer wall of fruit, it is called as pericarp. So don't confuse perisperm and pericarp. The pericarp is nothing but outer wall of fruit. The perisperm is nothing but that is remnant of nucleus in a seed. So these pericarp, it consists of 3 layers, that is epicarp, mesocarp, endocarp. So as you seen in the just a mango seed, it is the epicarp, outer covering layer. It is yellow colored or green in color. While it is young, green in color. While it is ripened, it is yellow colored or different color nature. It is the endocarp, where there is presence of inside that presence of seed. So in between endocarp and epicarp, the presence of mesocarp, it is a pulpy pod. So that is mesocarp. All these 3 layers, which together called pericarp. So as these epicarp, endocarp, mesocarp are the pericarp layers is of different in different fruits. For example, while you take in a fleshy fruit like tomato, that is outer epicarp and inner totally that is pulpy pod. Only the endocarp, that is only 2 layers. While you take a drupe as the fruit of that is mango or coconut, in these the pericarp is 3 layers. So already we discussed in previous class, that is classification of fruits, that is simple fruits, aggregate fruits, multiple fruits. Simple fruits, aggregate fruits and multiple fruits. In simple fruits, once again 2 types. So already we discussed the classification of these. Fruits categorized 3 types: simple fruits, aggregate fruits and multiple fruits. The simple fruits, which once again categorized 2 types, that is fleshy fruits and dry fruits. The dry fruits, once again, the 2 types based on dehiscence. Indehiscence, dehiscent fruits. Indehiscent fruits and dehiscent fruits. Indehiscent fruit means the fruit is not opened, like walnut or that is groundnut. Those are indehiscent fruits. So dehiscent fruits, it is nothing but, so after completely mature, the fruit is slowly opened. The fruit is slowly opened. So like certain bean, that is pulses fruit. The bean, it is open. The lady's finger, it is after matured, it is slowly open. That is datura. Datura, it is also slowly open. So based on opening, these dehiscent fruits are once again the different types. That is laculicidal, septicidal, septifragal. So depend upon that is the breakdown nature or dehiscent nature. So aggregate fruits. Here the fruits are formed by single flower but multicarpellary apocarpous ovary. Multicarpellary apocarpous ovary which developed as aggregate fruit. Example Aristolochia. As multiple fruits are nothing but there is fruit formed by entire inflorescence, either spike inflorescence or catkin inflorescence or spadix inflorescence. So like the entire inflorescence which developed as a fruit. For example, a custard apple, jackfruit. So which consists number of fruits which are number of fruits which are found present in the same fruit. So that is multiple fruits. So like the fruits are categorized like different types. So the fruit is once again divided as a two types, basically two types. That is false fruits, true fruits. False fruits and true fruit. As in false fruit, see as the apple or strawberry, that is fruit form by thalamus. The fruit formed by thalamus and all other part. Other floral parts. So in true fruit, it is nothing but fruit formed by ovary. So other thalamus, other floral parts is nothing but that is ovary. Means the ovary along with the thalamus which is present, the fruit is formed in false fruit. So see, it is the apple, it is a false fruit. This portion, it is completely, it is the thalamus part, it is the ovary part. Thalamus part and even the ovary part, entire it is leads to, it can take part in formation of fruit, that is a false fruit. So even in the, that is strawberry. So true fruits, the fruit formed by the only ovary. So ovary part is only takes place by formation of fruit. That is what we have, that fruits are called as true fruits. So after the seeds are formed, after fertilization of ovules, means the seeds offer certain advantages. Seeds offer advantages to angiospermic plant. So last time or before last time there is a question, name the advantages of the seeds towards the angiospermic plant. So what is the seeds? What are the advantages provided by these seeds towards angiospermic plants? It's very important. So here the reproduction, that is sexual reproduction or the reproduction such as pollination, reproduction process such as pollination and fertilization. So the pollination process and fertilization process are independent of water. So pollination and ಕೆಲವೊಂದು plants ಅಲ್ಲಿ ಮಾತ್ರ water ಬೇಕು. So the plants which are lived in aquatic condition, that is hydrophilic plants. But in most such terrestrial plants, the water is need not for pollination. Water is also need not for their fertilization. So as in bryophytes, they need water for their fertilization. But except those, the water is independent. That is, pollination and fertilization process are independent of water. Those are not water dependable. So, but water is required for seed formation. That is, seed formation is depend on water. So as without water in the fruit, how the seed is formed? That seed is very smaller in size. As like the seed formation is depend on, that is, water. So it is one of the advantage of seed towards the angiospermic plants. As after the seeds contain reserved food material, or seeds contain sufficient food materials. Sufficient food nutrients, as the seeds are used as, that is, food, the source of food. Seeds have better strategy for seed dispersal. For example, certain seeds which having a small cotton thread-like structures, as in, that is, cotton. The presence of those, the seeds are easily dispersed to the different area. So while the better strategies present in such seeds, the seeds dispersed to new area, that is, new species originated in new areas, that is, seeds dispersed to new area to colonize new species. So as which adopted the better strategies for the dispersal nature, hence those dispersal nature, it helps to the new species colonized in the new area. So after the seed coat protect the embryo. As we know that the integuments of ovule which developed as seed coat, that entire seed coat which completely protect the embryo. So even that is the seed is formed by sexual reproduction. Seed is formed by sexual reproduction. Due to sexual reproduction, there is combining of genetic material. The recombination is occurred at the time of meiosis, at the time of formation of gametes, as the seeds which are formed by sexual reproduction. Hence, the recombination of genes helps to arising new characters or arising variation. So like these are the different advantages offered by seeds towards the angiospermic plant. So seeds are which are formed after fertilization of ovules. That is, we know that those seeds in which dehydration is the thing what dried nature of that seed. That is presence of moisture only, that is 10 to 15% of moisture present in the dry seeds. Here 10 to 15% of moisture present in the dry seeds, it helps to the seed present in dormancy condition. Hence, the dehydration and dormancy is a very, very important process for which helps to storage of seeds. If the process dehydration is not occurred, the seed is not viable in long duration. If the dehydration is occurred, such seeds which is viable, it is little bit long duration. Because even though in the dry seeds the embryo is having viable nature, the embryo is viable in dry seeds. So if the embryo is viability, that viability of seed is depend upon this embryo viable nature. So it is depend upon the moisture content present in the seed. So once you assume these two processes are not occurred in the seeds, a mature seed produced by a plant, it is suddenly we are used as a food or different. Suddenly we are used for culturing of new crop. So otherwise, these processes are not occurred means such seeds are not using along there. Such seeds are not used in the next season. Next season ಅಲ್ಲಿ ಯೂಸ್ ಮಾಡಲಿಕ್ಕೂ ಹೆಲ್ಪ್ ಆಗುತ್ತೆ. ಸೊ ಕಂಟಿನ್ಯೂಸ್ ಆಗಿ ಸ್ಟೋರೇಜ್ ಮಾಡಿಕೊಂಡು ಫುಡ್ ಆಗಿ ಯೂಸ್ ಮಾಡ್ಕೊಳ್ಳಿಕ್ಕೂ ಯೂಸ್ ಆಗುತ್ತೆ. Like the viability of such seeds is not same in all seeds, but different seeds which are having different viability. So like as we know that a rice which is used 2 or 3 years, so more than that it is that is little bit decomposed. A fleshy fruit is not used long duration. A dry fruit, it is used in long duration due to dryness, due to less moisture content. As depend upon the viability of seed. So one of the oldest seed is lupin seed. It is Lupinus arcticus. The Lupinus arcticus seed, which is excavated in Arctic tundra. So these lupinus seed, which is viable nearly 10,000 years. 10,000 years. So these lupinus seed, which is excavated in 1954. In 1954, the lupinus seed is excavated in Arctic tundra region. So, but this seed, it is germinated in even 1966. So another one recent record of this viability of seed, that is in recent record or recent viable seed, that is date palm seed. Date palm seed. It is Phoenix dactylifera. Phoenix dactylifera seed. That is, it is excavated near Dead Sea. That is King's Herald Palace. King's Herald Palace. So this Dead Sea, it is present in Israel. So especially the area called Masad. In Masad area, that is the Israel, there is presence of Dead Sea. The Dead Sea present east to Jordan. That is west to Israel. So ಈ area ಇರತಕ್ಕಂತ Dead Sea there is archaeological excavation occurred. So in that excavation there is a recent record of date palm. So it is 2000 years viable, 2000 years old seed. 2000 years old seed. It is excavated by archaeological excavation near Dead Sea. So Dead Sea it is also nothing but a salt sea. It is totally salty nature. So where there is excavated. So but this seed, it is germinated in 2015. 2015 alli germinate ಮಾಡಿ. So even though more than 2000 years, it is germinated, it flowered, it given fruits. So it is the viable nature of that seed. So the last topic, apomixis and polyembryony. Apomixis and polyembryony. So it is apomixis. So here, apo, it is nothing but away from. Mixis, It is nothing but fusion of gametes. So here, in most of the angiospermic plants, the seed formed after fertilization the ovules which developed as seed. But in these, that is absence of meiosis and syngamy. That is, there is no meiosis, there is no syngamy, but formation of viable seed. That is, formation of seed without fertilization, such phenomenon called apomixis. Apomixis is nothing but formation of seed without fertilization. That is, absence of meiosis, absence of syngamy, even the formation of viable embryo. Viable embryo is formed in the seed. Such seeds are called as apomictic seeds. So one thing you discussed in the hybrid seed formation. So these apomixis is present in the hybrid seed. It is little bit advantageous to whole farmers, because today what we are used, the hybrid seeds, the hybrid seed formation process is little bit more expenditure. So that's why every year a farmer went to market who purchased the hybrid seed to grow their crop. When, like in the hybrid seeds are formed by these apomixis process, there is no need of purchasing every year. What he grown the seeds, those seeds are used to raising the crop in next year or next year. So it is not occurred. These testings are now also it is continuous to raising hybrid seeds with the apomixis process, but it is not completely successful. So this process is continued. So these, the apomixis which is present in certain species, that is example, certain species of Asteraceae family. So Asteraceae family, all members are L1 species marker. So and most of the grasses, grass family, that is Poaceae. So both in which the seeds which are formed without fertilization, those are called apomixis. Such seeds called apomictic seeds. A seeds are now apomictic seeds and telecaritive. Apomictic seeds. So apomixis is nothing but formation of seed without fertilization. Such process called apomixis. So next, polyembryony. So name also indicates the last topic, it is polyembryony. Poly is nothing but many. Embryony, formation of embryo. So polyembryony is nothing but the process of formation of more than one embryo in a seed. In a single seed, there is formation of more than one embryo. more than one embryo. Such phenomenon what we are called as polyembryony. So these polyembryony, it is common in orange and lemon seeds. So just you squeeze the lemon seed or orange seed, there is presence of more than one embryo, but most of those embryos are sterile except one or two. So in more cases, only one embryo is completely matured. So remaining are either degenerate or they are not completely developed. So these polyembryony, it is first reported by Anton van Leeuwenhoek in 1790. It is first reported by Anton van Leeuwenhoek in orange seeds. So what is the causes for these polyembryony? So just we discussed these causes. So there is presence of embryo sac. So surrounding there is presence of nucellus in the ovule. If sometime the nucellus cells may enter into embryo sac, they are developed as embryo. Certain cells of nucellus may enter into embryo sac, they can take part in formation of embryo. So as it is formation of more than one embryo. Sometimes the embryo sac consists of synergid or antipodals which are also fertilized with the entry of more pollen tubes, during which these are also developed as embryo, but maybe most of them are sterile nature. Or otherwise, certain sporophytic cells of embryo sac is also developed as embryos. By these causes, maybe polyembryony is occurred, that is, formation of more than one embryo in a single seed. Such phenomenon what we are called as that polyembryony. It is ending in this, end the second chapter. We continue in the next class, the next chapter. Thank you.